found this insulin degrading enzyme, which is a zinc metalloprotease, and the interesting thing is it's capable of degrading all those things, but especially in the brain, it is the one mostly responsible for degrading the amyloid beta, which is very related to the Alzheimer's disease. There's some experiments in rats that show that if you can somehow remove the amyloid beta plaques, rats recover recover memory and memory skills. So the idea is if you make this enzyme work better, then you can have a better drug that actually targets Alzheimer's disease cells. So this is a big system, right? 4 domains and opens and closed like a shell. There's not going to go to full details of the simulations here, but here's the active site. The things that it needs to open, and then the peptide gets in, and then it closes, and degrades the peptide, opens again, and the peptide leaves. And it also has an ATP binding site that's around, the ATP binding site, this arrow is out of the place. ATP binding site, it's around here, and then there's this, the active site is a kind of a small distance from there. And what we found is that, well, there's some things I didn't tell you. In the presence of ATP, in the presence of ATP, this enzyme gets less active. It loses activity in the presence of ATP. So what we found is that when amyloid beta is in it, it binds to ATP, and it keeps it keeps the enzyme in a closed state. It doesn't open anymore. So and in the presence of it, so this is the simulation that was seeing how this measured the interaction of the amyloid beta with ATP. So it keeps the enzyme in a closed conformation, so it doesn't open for to release the products. And well, we also did some QRM simulations of the system here, which is cut here. But it was a full 2D umbrella sampling here for this that connects those states. And he did this in the absence and in the presence of ATP. And you find that when ATP is present, there's a larger barrier here. So in the presence of ATP, it not only avoids the enzyme to open, but it also, the reaction stops working or works less efficiently. So basically, amyloid beta gets in and gets stuck in there. So the idea is, well, that opens the possibility of developing things that will interfere with ATP binding site on this enzyme, and then we would have something that makes the enzyme work on overdrive to clear the amyloid beta deposits in the brain. That was the whole idea. And just yesterday, I've seen some works coming out that's already trying to use this enzyme as a target for Alzheimer's disease treatment. Well, but the other thing is the one that I'm going to spend longer here is this dengue virus protease, which is a work that's just finished. We're just finishing. We're about to send it now. And the thing about dengue, and which it's kind of getting, it's losing a bit of momentum now, dengue, because Zika is in fashion right now. Everybody worries about Zika. But Zika and dengue viruses are very similar. They're from the same family, and they have lots of things in common. And just Zika affected the central nervous system harder than dengue, so they got more attention now. But this is just I got this 2 days ago. In the past 3 months, there's been dengue alerts in all these regions here in the world. And more specifically, this is especially interesting in Brazil. Right before this, everybody talks about Zika. What we noticed is this here is giving you telling the number of dengue cases in Brazil, and this dark line here is on 2014. You see, well, it's kind of regular thing, right? And in 2015, right before people start talking about Zika, there was a huge increase in the number of cases. So it went for, I didn't get the number here, but about I don't know, less than 30,000 cases of reported cases of dengue in 2014 on the 13th week. I'm just showing the 13th week of the year, okay, just up to the 13th week. And then in 2015, we already had 705,000 reported cases of dengue. So it was, everybody started talking. There was a 900% increase in cases of dengue here in Brazil, from one year to the other. And people started getting worried about this. And after that is when the Zika appeared, or not really appeared. Well, after that was the Zika worries came. So what happened is the vector that transmits dengue is the same that transmits Zika. So when Zika was already in Brazil for a while, but then when the number of cases of dengue went up, of course, number of Zika cases also went up. It was the same vector. And then, so this, keeping the proportionality here, the same increase happened to Zika cases. And well, this is not the whole story. In 2016, so this is until the same week in 2016, and that's why I'm keeping the plot to the 13th week. We already had another increase at about 10% over the 900% we already had in cases of dengue. So even though we found out last year that things are problematic, we didn't do much to solve it, and we still, the number of cases still went up. And if you didn't have dengue yet, good for you. You get a lot of get some high fever, headaches, you know. And by the way, there was an estimate that said it was more than $9 million a year that's lost because of dengue cases. And the thing is, since there's no real treatment for it, the only thing that we need, the only thing that's being done for that in practice is the government programs to control the spread of the mosquito, right? So this is all we've been doing, and we all know that those government programs have some years they work, some years they don't. Now you've seen things like this. This happens. Okay, so you cannot just depend on what the government's going to do. Okay, so what happens if there's a great, at least back in Recife, in a family, usually if there's someone who didn't get dengue yet, he will get. It's basically everyone there has it at some point. Keep in mind that only about a third of the cases do show symptoms. So some people might have dengue, might transmit dengue, but might not be symptomatic. And the same thing with Zika is even worse. It's about 1 in 5 cases show symptoms, so it's even worse in this direction. But anyway, if you already have dengue, the only treatment that is, well, there's no drug for it. Okay, so all you have to do is take Tylenol and water. That's the dengue treatment. And if you get dengue a second time, it gets even worse because there's some specific interactions there. It can lead to some condition called hemorrhagic dengue fever or dengue shock syndrome that can lead to death. But all you can do is just take Tylenol and rest. So our interest in dengue started from this because we wanted to, when we came to Brazil, we wanted to work with something that affects locally. And in the case of dengue, the story of dengue is that the way the viruses work, and you've seen with Sergio's talk here, the Zika, the image of the Zika virus, dengue is about the same. Also has all those proteins which are about the same proteins that the Zika virus has. And the interesting thing with the virus produces a polyprotein. It has a polyprotein. Well, actually, the RNA of the virus will be translated into a polyprotein, and this polyprotein needs to be cut at some places for the virus to work. And what we found out is that some specific positions here, there are cut by a specific enzyme from the virus called the NS3 protease, which uses this NS to be as a cofactor to work. Okay. And it not only cuts the enzyme here, but NS3 protease also cuts some enzyme on the host that is responsible for the, let's say, the alarm system. So it not only works for the virus reproduction, but also shuts off the alarm on the host cell. Okay, so this is the first structure of the NS2B, NS3 pro, which is this protease here. It's a kind of simple protease, and here's the enzymes of the active site. That's a serine protease, kind of like trypsin. And the mechanism, the suggested mechanism is, it involves, this is it will break a peptide bond here. Remember, it has to separate polypeptides, so this work is breaking a peptide bond. So the serine attacks the peptide bond here. And I should say this enzyme also has another domain that's called the helicase. We're not going to show you anything about this one here, but there's also some work about some helicase inhibitors, which is it's a domain that works on unwinding the RNA so it can be processed by the cell. Okay? So it's another target. And the mechanism here, to give you some more details, what is believed is that you need this protein to be transferred to this nitrogen here on the histidine that activates the serine, and then serine can attack you. There's been some discussion in the literature about the paper, the role of this residue here on taking this protein, so this whole process can happen. And then as this happens, as this protein is transferred in and the serine attacks the substrate, there's a negative charge formed here, and usually the serine protease enzymes, they need to have some residues close by that complete what's called an oxyanion hole, and that stabilizes this oxygen here. Unfortunately, nobody has seen the oxyanion hole in this specific serine protease. Nobody says why, but in the crystal structure, it's not identified, and some other simulation works don't identify the residues that make the oxyanion hole. And then this hydrogen that came from serine is now transferred to this nitrogen here, and as it transfers, the bond breaks. The peptide bond breaks, and then in the next step, a water molecule comes here and restores the system. So to simulate that, we wanted to make the simulation the whole enzyme environment, and we had a model peptide, a model substrate, which is basically there was a study in around 4 years ago, where the authors had a large number of peptide models, and they actually did it. It was like an experimental work, and they tried to find which peptide combinations this enzyme is more efficient with. So we got the one that's very efficient, this small peptide here. So this is the bond that's going to be broken. So this one was the whole QM system. Okay, they involved all the catalytic triad atoms and a couple of atoms more to the right, more to the left, so that we don't cut the peptide bond. And this is the substrate. So there was a total of 140, actually it should be 142 atoms here. So it's 48, it should be 42. We use this PDDG PM3 Hamiltonian, which has some good results for proton transfer reactions. the reaction was actually separated in two steps because we wanted to see, go back here, we wanted to see two things. We wanted to see how the activation of the serine works, and then the attack of serine on the substrate. Okay, so it's basically a two-step thing. For the first step, we had to use, there are some more, I'm going to show you that there are more things involved. So it had to be a full 2D umbrella sampling, so it was 120 windows. And the other one was simpler, 16 windows was enough, and had 35. It's not too much if you think about regular molecular dynamics time, but when you're doing quantum mechanics, 35 picoseconds sampling is, well, it takes a while, okay, to do this, and you do this in 136 times, and that's just the ones that work. It's just, anyone who runs simulations here know how much time you lose with things that don't work. So this is showing you the results of this first part, and we found some interesting things here. And we'll start from the enzyme substrate complex here, because there's a first transition state, and then it really does form this proton transfer intermediate here, which what's happening here is that as you shorten this D1 distance, which is the distance for the proton transfer from serine to histidine, and we're looking if this transfer is concerted with the attack here. What it turns out is that the attack cannot happen until the proton transfer is basically transferred to the histidine. And this only happens, and I should have, but I don't have a plot here, but it only happens when this proton is transferred. Okay, so we need this one to transfer to activate the histidine that activates the serine. It's a kind of domino effect here. And then only after this proton is transferred, then serine can attack. We did a full scan of the surface here, so there's no way it could be either concerted or you had a serine attack first. And then, well, you can look at the energies of those states. It had a big activation. The first step is a kind of big activation barrier, which is about the same ballpark of all serine protease around the 20s of kcal per mole. And we can also look at the charges here in the state is pretty consistent with the mechanism. I'm not going into the details here of the charges, but you can see how this, during the transfer, this thing gets more basic, so it takes the hydrogen here, and then the serine charge needed to the attack of the serine here. And one thing that's kind of interesting that showed up in this system, we didn't force it with, is that some water molecules started to showing up around this oxygen here and stabilize the charges. So what it's telling us is that the so-called oxyanion hole that in most enzymes is formed by other residues that are around it. In this case, it's formed by water molecules that come to stabilize this oxygen, which is reasonable because in this enzyme, the active site is pretty exposed to the water, but it's a suggestion that hasn't been talked about before, not on this one. We can see clearly that in the beginning you have, in most of the frames, well, this is after we got the mechanism, then we got some more detailed sampling on each of the states, each of the important states here. So those are results of another 50 picosecond sampling on fixing those states. We can see that there's a lot of a lot of the frames have only one water molecule around the oxygen here, and then as you go and you form the transition state, so this thing's getting closer, you suddenly have a jump of two water molecules always at least two water molecules always present around the oxygen, and when you form the intermediate that has a negative charge here, now you have even three water molecules around the oxygen. So the oxygen hole in this one is formed by water. This is doing the work there. So you can see the mechanism here. Well, I should say that this is not a real movie of, well, let's say, of this thing happening. We had a whole surface. Okay? We scanned the whole surface. What we did is we got frames from the lowest energy path and just build them into a movie here. Oh, not what I wanted. So we're able to see here, see the first proton transfer is kind of happening here, and then the histidine is activated, and then finally, with time, histidine will approach the hydrogen from serine, so we'll take the hydrogen off, and you see that this system is always kept in this direction here. And now it's getting to the hydrogen, and only after it takes the hydrogen, it will be possible for the attack of serine here. It takes a bit because it's a long path here, and it's about all the windows that are in the path. And then after that, it will be able to attack. You see the water molecules already. This water molecule was traveling all around. If you notice in the beginning, now with time it gets more and more present around this oxygen here. You see it comes back all the time. There's another water molecule. I don't know if it's going to show up here. It comes from below here. So as the serine is losing its hydrogen, and then it will attack the carbon. It does take long. There's a bit of sampling on the intermediate state as well here, so that's why it stays there for a while. No, that's the thing. That's the next step in the work is to make the water QM as well. But at this point, the water is just a MM water. It's a regular MM water. You see, for a long time, it stays there, and then at some point, it should start attacking here. Yeah, now it's coming down. So we're controlling only these two distances, okay? This hydrogen distance and this attack here. So we'll attack, and there you see the formation of the tetrahedral intermediate. On this point here, the only thing that's left now is to break the bond. And to break the bond, what we did, we tried to drive this hydrogen to the nitrogen here to make the bond break. You see the water molecule doesn't leave here anymore. It's kind of stuck there, stabilizing the oxygen charge. Now for the second step, we did about the same thing, but now all we did is to drive this hydrogen that now was in the nitrogen, to drive it towards the nitrogen. And this second step has a lower barrier, okay, but then it stabilizes a lot later. So it shows that the rate determining step must be the first one. Well, just the charges are consistent. I'm not going to go into the charges here. And then as you go, as the reaction happens and the peptide bond breaks, then there's no need for those water molecules around the substrate anymore. They go away, and now we go back to a regular distribution, just a water coming by there every once in a while. So now you see that just as we drive this proton here, the bond breaks naturally. We didn't have to control the bond breaking at all. So this was a simple 1D umbrella. So as it connects there, the bond instantly breaks, and then you have the products here, and you get the ACR enzyme intermediate. So yes, after the no. Well, the thing is that when you're choosing the structures to start each simulation, you have to we're looking for one structure from the end of the previous simulations that had the best distance between this hydrogen and this nitrogen. Where's the nitrogen? Yeah, here. Which had the best distance between those two. Okay, and this one happened to still have this complex performance. Okay, we looked at one that was closest so we could save a little bit on the umbrella. Right? Anyway, those are the results of this little thing. We have some other people working in the lab with the idea of getting, scanning a database and some ensemble docking the structures that we take from those simulations that will allow us to suggest. There are some experimental collaborators that will test any hints we can give them, and maybe we can find something that happens to help whoever already got dengue. Okay? Well, this is just not going to talk. I've already talked over my time here, so I just want to thank some things involved here. I had to thank the campus in Brazil that gives the scholarship to the students to do it. Most of the money actually came from CEPID, the state foundation, and some of the calculations running in CEPID that we have in Recife. I have to thank Munir and the organizers to call us here, and I'll thank you for not falling asleep here in the, at least most of you, not falling asleep during the talk here. I can take any questions you have. Here? Oh, in the first step, we had two reaction coordinates. It wasn't a combined reaction coordinate. It was really, we were really controlling this proton transfer in the first step, and at the same time, this attack. Okay, so there was a real two-dimensional sampling. And on the second step, we were controlling just this attack, just one dimension. Yeah, it varies depending on in each window. I don't remember the values now, but some windows it had to be pretty high, especially when you're sampling the transition states. Had to be pretty high. I could show you the numbers. I just don't have them in my head now. It reached 1,000 or something. Yes. Yeah. No, I'm only controlling this distance here. As it approaches, everything else is, they can move around as much as they want. No, no. This step was actually pretty easy. If you look at the energies involved here are not very much. Like it took 5 kcal per mole to break. So we didn't have to force much here. The real challenge was the first step. much of a difference if the umbrella is done correctly. If you have good sampling in each of the points there shouldn't be much of a difference. You would see more hysteresis if you pull it too fast, which could happen in Jarzynski case. If you pull it too fast, then there's a large hysteresis. But in case of umbrella, you don't just, it's slow. You put the system in a position, and then you thermalize it, and then you start some production, and then you only use the last steps, and so there's the whole control that reduces this. I'm not able to answer this question. I'm really unable to answer this question now because we didn't see this, and I didn't look into other. In this case, I can tell you that we didn't see this. The two cases that I showed here, the other one was with Carlos. There's also the same slow control of the coordinates, so that we avoid even worrying about this. Not in this case. There's not enough. There's no waters around here. We look at it. We had before. Yeah. Before we it's not right. But before we actually run the QMMM simulations, we had some really long MD simulations on those systems, right? More than 300 nanoseconds or so. And there's no water molecules around here that could be involved in the proton transfer. The water molecules that show up is actually closer to the substrate because it's the more solvent exposing part. So we considered this possibility. We looked for water molecules there extensively. So we spent some time looking for the density of water molecules around it. So there's more stuff that I'm not showing you. We didn't find any water here that could be involved.